So if you think back at the very beginning of this unit, one of the things we talked about was that scientists from China who genetically, so the terms we might use were genetically engineered or genetically modified some embryos. And his work attempted to make those two twins resistant to HIV virus that's sexually transmitted. And so we talked a little bit at that time about is that should we be able to modify the genes, the DNA of humans? When is it appropriate? When is it not appropriate? So we're going to talk today about some of these things. So there's a term we use, a general term for scientists kind of manipulating and working with DNA. We call that genetic engineering. It's a big field. It's a growing field of study, especially now. In the last 20 or 30 years, scientists have learned a great deal about DNA, how to work with it, how to alter it, how to take genes from one organism and put them into another, how to actually edit the genes in a person's body, in certain cells in their body. So science has come a long, long way. And really, a large part of the future of medicine is going to be related to genetics, going to be related to DNA and how we can work with it. And so that's what we're talking about, some of those topics here. So this term, genetic engineering, it's a general term, but it's sometimes used to mean altering the genetic code of an organism. So we already talked about in the last few days. The genetic code is a series of letters that's in our chromosomes, right? In all of our chromosomes, if we were to look at the DNA, it's a whole bunch of letters, A's, T's, G's, and C's. Millions of them. And the sequence of those letters make us who we are. Those are our traits. The sequence of those letters is why I look the way I do. It's how my body produces proteins, how my organs function. Those things are coded for in those simple four-letter alphabet. But scientists have learned how to change some of those letters. Scientists have learned how you could take a gene from one organism and put it into another. A gene even from a different species can be added into the genetic code of another. And so we're going to talk about the steps of this a little bit. And so first off, some things to know. We can extract the DNA from a cell pretty easily. You could use chemicals to sort of break open the nucleus. You can purify so you just have the DNA. You can sort of produce more copies of that DNA. And then what scientists do, if they want to take a gene from one organism and put it into another, they have to basically cut it out. It's like using scissors almost. You cut out the gene you are interested in, and then they paste it into the DNA of another organism. An important term for you to know is this term, restriction enzyme. So it's an enzyme, which means it's made of a protein that helps some chemical reaction take place. Restriction enzymes are how scientists cut DNA. DNA is really small. You don't use like scissors or scalpel or even a microscope to cut the DNA molecule. You actually use these special enzymes called restriction enzymes. And they cut the DNA at a specific sequence of bases, AAGC, for example. You could get a restriction enzyme that anytime it sees that sequence, it's going to cut the DNA. And so scientists can use these restriction enzymes like scissors. They cut out a gene from one organism. They can then use restriction enzymes to cut the DNA of another organism and insert the gene into that DNA. Okay. And so this has happened in a couple different ways. So we know from when we talked about homeostasis, people that have diabetes whose pancreas doesn't produce insulin, they need to administer insulin to themselves. Okay, so it doesn't come from their own pancreas. Well, where does it come from? Back 30, 40, 50 years ago, they extracted insulin from pigs, from the pancreas of pigs that had been slaughtered for meat, and they could extract insulin from it. And that's what people with diabetes would use. And it worked okay. Some people didn't work great for, some people are allergic. But since then, since we've learned how to transfer genes from one organism to another, scientists have a different way of making insulin. It's kind of shown here. Now, this example says human growth hormone. It's also done for human growth hormone. But we talked a lot about diabetes, so I'm going to use insulin as an example. So, in my cells, for example, I have, we all have genes for how to produce insulin, right? Insulin is just a protein. So somewhere in our DNA, there's a sequence of A's, T's, G's, and C's that tell our body how to make insulin. So scientists can take a cell from a person, find the insulin producing gene, use restriction enzymes, cut it out. Okay, that's this step. And then what they did is they take bacteria DNA, cut it open, and they paste in that insulin gene into the bacteria DNA. And they put it back into bacteria. So we see here we have the insulin gene. It's put into bacteria, okay? And then it's put back into a bacteria cell. You can see the purple represents the insulin gene. So now that this code, this recipe for how to make insulin, it's in this bacteria. So guess what that bacteria can now do?
Split, copy it.
Okay, it can reproduce. And when it copies and there's more bacteria, they all have that gene in them.
Yeah.
So what do you think those bacteria can do if you grow them?
Help the body.
By, what is this insulin gene going to do?
It's going to grow.
It's going to allow these bacteria, they couldn't do it before. They don't have an insulin gene. Bacteria don't need insulin. They don't use insulin. But since scientists put an insulin gene into this bacteria, it now produces insulin. And they grow these bacteria, actually, in big vats. And then they're all producing insulin. They can extract that insulin, but it is human insulin, right? Because this is the code. This is the instructions on how to make human insulin, and they put it into bacteria. So now that's how modern insulin comes from genetically engineered bacteria that they can extract this insulin from. And it's identical to human insulin, really. They can do it with other hormones as well. This is where human growth hormone. Sometimes people have a deficiency of human growth hormone and they don't grow to a normal height and they need injections as our children so they can grow to a more normal height. To get that human growth hormone, it's made through the same process. Inserting the human gene for growth hormone into bacteria, growing them, and extracting the growth hormone. Mr. McCoy, anything else about insulin that you know about genetically?
Would this be the same thing as what they call synthetic insulin, or would that be?
I don't know. I mean, because I'm not sure. Yeah, because some hormones and proteins and other things, like you can basically create them in a lab, and maybe is that you can get synthetic?
I think that's what they're using now is synthetic.
Okay.
I want to say.
I'll have to do some more research on it.
I'll have to check that out too.
Yeah, interesting.
I don't know if that was the same thing.
I don't know, maybe it's just a different term for this. Yeah, I don't know. So here's some other examples. Did you have a question, Andrew?
Oh, sorry.
No.
I saw a Hobart education T-shirt because my mom used to go to Hobart.
My daughter goes to Hobart too. So here's some other examples of this. So we call those when we take a gene from one organism, put it in another, they're called transgenic organisms. Here's some examples. I already talked about this, the bacteria that we put human genes in. You could add, you can add extra genes. Go ahead, Mr. McCoy.
Sorry, it is synthetic and it was made using E. coli bacteria.
Okay. So yeah, that's like the same.
1978.
Okay. So that's just the same term for what I was showing.
Okay.
And I think this slide it even shows. Yeah, these are E. coli bacteria. So they have genetically engineered cattle, beef cattle, and put a gene that allows them to produce extra growth hormone. And guess what those cows do?
Make more milk.
They grow larger because they have extra growth hormone. They produce more meat.
Could you put animal cells in humans?
You could buy basically any cell from any organism in any other organism.
I want to have a monkey DNA. Gorilla. So I can like.
So they can take genes from how to produce medicine, so they are made in the milk of animals. That's another example. They can take genes in plants. This has been done. They've made a special type of rice called golden rice, and they put a gene that produces more vitamin A in this golden rice. And vitamin A is important in the development of vision. And people that are deficient in vitamin A can suffer from serious vision problems or even blindness. And so when people don't have enough food, don't have enough vitamin A, that's a serious problem. So they genetically engineered rice so that it has extra vitamin A that people can have that helps provide enough for them. So there's lots of examples of this. They've taken genes that produce insecticides and put them into crops so that now these corn plants, for example, produce these chemicals that prevent insects from feeding on them. But again, scientists did this in the lab. Sometimes you may hear of this term GMOs, genetically modified organisms. Like sometimes foods are labeled, contains GMOs or does not contain. So genetically modified organisms, some people have the opinion that we should not be doing this, that we shouldn't be taking genes from one organism and putting them into another.
Yeah, they say it's against God or whatever.
They say that it's not natural. Some people say, well, what if we take some gene, what if somebody's allergic to something and now it's in a common food? What if that happens?
And we all die.
Sometimes, people just feel that it's not ethical to be cutting and pasting genes from one organism to another.
Why not make superhumans? Like, that'd be cool, right?
Well, and
Like, what if we take monkey, gorilla, and crocodile DNA and bodybuilder DNA?
well, what's somewhat, I think, a little bit more interesting is this idea. So now this is a newer tool. So we've been able to cut and paste these genes from one organism to another for a long time. But now, in the last 10 years or so, scientists have a new tool that they can basically edit the DNA inside of a living organism in certain parts of that. And we're going to watch a video that talks about some of this. So basically, they can edit genes inside of a person's body. It's called CRISPR-Cas9 is the name of it. It doesn't matter for your purposes. But basically, they could take, if a person has a genetic disease, scientists are close to having the ability to take a healthy gene for that trait and replacing the defective gene. For example, when I was in high school, I had a friend, she had cystic fibrosis. Cystic fibrosis is a genetic disease, and it causes mucus in a person's lungs to become too thick. It's caused by just one of those DNA bases, one of the A's or G's or C's or G's being incorrect.
Oh.
And so my friend in high school, she had this disease. She was born with that. She inherited it from her parents, and she had constant lung infections and so forth. At the time, there was no treatment. It was a genetic disease. It's in all of your cells. You can't cure it. You could treat the symptoms and things like that, but she was born with it. She actually died in her early 20s from cystic fibrosis. But with this technology of genetic medicine based on CRISPR-Cas9, we're getting in the realm of she may have, if she were born today, maybe in the next 10 years, they could have given her a treatment, changed this gene in her lung cells, changed that unhealthy version that gave her cystic fibrosis to the correct version, and basically fixed this genetic problem in her lungs, and maybe she would have been healthy. So we're getting there, very close to where some of these diseases that are caused by a single identifiable genetic mutation, we may be able to correct those things. And so that's the future of genetic medicine. But again, and there's a question of some people say, well, should we be editing human DNA?
Well, yeah, we can clone people.
And some people feel, some people would feel, yeah, if we could correct a serious disease in a person, we certainly should do that. But the question comes, well, what do we consider a serious? What if we could change to make people taller, right? Or what if we could select for eye color or hair color in our children? Should we do that? Should we allow for that? So the question is, I don't know. What about if it is expensive? This type of treatment will be expensive. What does it mean if only people that are wealthy can correct certain genetic traits or can select for certain genetic traits? What does that mean about the ethics of whether or not we should do this? So there's lots of interesting questions to think about, ethical questions regarding.
We can clone people.
We can clone people.
Well, not people.
We can clone people.
We can.
We haven't yet that we know
Clone
of.
like animals, like salamanders.
So cloning is a separate topic. So cloning is making a genetic copy of an organism. The first mammal to be cloned was a sheep.
Yeah, we get salamanders and stuff.
Yeah. The first cloned mammal was a sheep. Her name was Dolly.
Uh-huh.
Cloning is a way to make basically a genetic copy of an organism. And the way cloning works, it's really not that complicated. So, the way cloning works is you need an egg cell from a female animal. You get rid of the nucleus. You basically suck it out, throw it away. So you just have an empty egg cell. And then you need a full nucleus from another cell. So let's say we want to clone Luke.
Yeah.
All right. So I need any cell from Luke's body. It could be a cheek cell, skin cell, cell from his hair. If we have one cell from Luke, inside of the nucleus of that cell has all of his DNA. Remember, every cell in our body has a full copy of the DNA, no matter where it is. So we could take a hair from Luke, extract the cell, take the nucleus out of that cell, and we put it into this empty egg cell. So now we have an egg cell, but the nucleus in that egg cell
It's mine.
came from Luke. It has all of Luke's DNA. If you zap that egg cell with a little bit of electricity, that causes it to start to divide through mitosis. It grows into 2 cells, and 4 cells,
Yes.
and 8. It starts to form an embryo.
Got sperm.
Yep, without any sperm cells. So, this is the little embryo. Once it reaches a certain size, you could implant it into a woman's uterus. She would be what we would call a surrogate mother. She would become pregnant, right? But the embryo growing inside of her uterus, it's not related to her. It doesn't have any of her DNA. The only DNA that's in that embryo is Luke's. And so she would be pregnant, fetus would grow, eventually she'd give birth. But when she gave birth, that would be a clone of Luke. Now, does that mean Luke and this little baby have the same mind? No, they're 2 different people. It's basically like you have identical twins, but they're not the same age anymore, right? And so that's how cloning works. All you need is a nucleus from a cell, an empty egg, and then you implant it. And that's what they did with Dolly. They took a nucleus from one type of sheep, put it in an empty egg cell of another sheep, shocked it, implanted it into a sheep. She was pregnant. She gave birth to cloned versions of Dolly. Scientists haven't yet cloned humans. So, I don't know why not. People have claimed to have cloned humans, but never provided evidence, genetic testing to say they actually did it. It's like internationally it's banned to clone humans. It's kind of interesting to think about though. Another interesting thing, a woman could use one of her own egg cells and one of her own nucleus from one of her own body cells and give birth to her own clone. Because, yeah, so, I don't know. Interesting ideas to think about.
I mean, I don't know what's wrong. Make a perfect version of yourself.
Well, it's not going to be any different, right? It's just got the same DNA as the person that was cloned. Now, what is commonly done is tissue cloning, where rather than making a whole person, they could take, say, liver cells. They could clone them, they grow them in a culture, and actually experiment on them. This is done commonly to help produce medications and do testing for diseases.
Instead of animal testing.
Yeah, instead of animal testing, instead of testing on a human, they can actually grow human tissue.
Why can't they use it to replace organs? That'd be like a good idea.
Yeah, that's probably something that is in the future. You can grow organs somewhat. Yeah, so that's probably something happening in the future. A couple other topics. This is called electrophoresis. Electrophoresis is something where you take a sample of DNA, you cut it with a restriction enzyme, remember, which chops up the DNA, and then you put it in this gelatin-like material, and you run electricity through it. This is the device, what it looks like. And when you do that, the DNA has a charge. It's attracted to the negatively charged part. And so the DNA starts moving through this gelatin. You run it for a certain amount of time, and then you stop it. Now, tiny pieces of DNA, small sections, can go far because they squeeze through all these holes very easily. Large parts of the DNA can't move as far. And so after you're done, you stain it, and you end up with a pattern of bands in this gel. This is the result of an electrophoresis. Okay? And this information can be used in a few different ways. Here's, again, an example. You cut up the DNA, you put it in these wells, add the electricity, the negatively charged DNA goes towards the positive end, and you have a pattern of bands. It's used often to match samples. For example, to determine paternity. If there's a question about who the father of a child is, DNA electrophoresis can be used to determine that. So here's an example. Here's a mother. Take a sample of her DNA. Chop it up with a restriction enzyme, put it through the gel, it forms this pattern. You could take the child, same thing, a sample of their DNA, chop it up, run it through the gel, this is the pattern. And then we have a few potential fathers. We do the same process with each of their DNA. And then you can compare the DNA in the child, the pattern of bands, and you can see where did those bands come from. So, for example, in the child, there's this band here. Did that come from the mother? No, she doesn't have a band there. It must have come from the father. Could it have come from this father?
No.
This?
No.
Yeah, there's one there. Or this father.
Yeah, that one there.
Okay, so let's continue. This band could have come from the mother. This one from the mother. How about this one?
No.
Could have come from him?
No.
No? Yes. And this one? No? Yes? Yes. This father all of these bands in the child match up with either this mother or this father. This bald guy is the father. So this can be used in crime scene investigation. If you have a sample of DNA from a crime scene, from a blood spatter, from cells that are left at the scene of a crime, they can do electrophoresis. And then if they have a sample from a possible suspect, they can match it up. And that's how DNA evidence can be used to help solve crimes. It's also been used to help exonerate or get people off of. So there's so DNA testing like this has only been around for about 30 years or so. There's this group, it's called the Innocence Project. What they do is they take people that are incarcerated, that are sometimes on death row, have the death penalty or life in prison, that maintain they're innocent of the crime. And if they were convicted of a crime before DNA testing was available, now they can actually go back, if there's still evidence from the trial, and actually test their DNA and see if it matches DNA from the crime scene. And sometimes they find that it does not. And so dozens of people that were in prison for life or had the death sentence have been released from prison based on the results of DNA evidence. It's actually a person in our area, Stephen Barnes, is a man that was convicted of the rape and murder of a girl in Whitesboro. A girl was murdered.
And raped.
Yep, I think she's a high school student.
Oh.
He was convicted of the crime. He always maintained that he didn't do this crime, that he was innocent. Eventually, in the maybe 10 years ago, 5 years ago, the Innocence Project worked with him. They took samples of his DNA. They had samples of DNA that were found on the victim. And when they compared them, they were not a match. He was actually released from prison, and now he's free. They never found the actual murderer.
Well, that's scary.
Yeah, but again, this is an example of DNA evidence being used in sort of legal cases.
Did the police ever give an apology?
He won a settlement of millions of dollars for, I don't know, it was a wrongful imprisonment, something like that.
Yeah, this is what I should say. If you ever get wrongly accused, the police should always apologize.
Yeah.
I mean, I feel like it'd be more than an apology.
Well, he did get, I forget what the amount of money he got. It was
Probably
in several millions.
Probably $1 billion.
How long was
And
he in
he was
jail?
in prison for 25 years or something.
So his credit score is in the toilet.
Yeah. Yes.
But still, he's got resources, shall we
Yeah,
Yeah.
but I would guess if you were to ask him, he would give all that money back if he could have 25 years of his life.
Yeah, imagine
freedom.
you're like,
I don't know. Me personally, I wouldn't.
No amount of money ever bought a
really?
second
You ever see what's
time.
inside jail? I've seen Scared Straight Man. It ain't nice.
Any questions about any of the things we talked about in terms of genetic engineering, genetic technology, cloning?
I like it.
5 seconds, y'all talk about murder?
Yeah.
I mean, we can talk about other things with
I mean,
DNA.
I'll show you some. Well,
Have you ever seen the Maury show? They use this type of stuff.
yeah, it's like who's the father? Yeah, I know.
You are the father. In the case of 21-year-old Janet,